So we know that living entities have genetic material and are able to replicate, so are viruses living entities?
But what does this actually have to do with the real world?
Actually, I wrote a study about MS, EBV, and CMV.
What are those?
To explain that, I think I'm going to need a little bit of help.
MS, or multiple sclerosis, is a disease of the central nervous system that affects more than 2 million people worldwide. It's an autoimmune disease that is often diagnosed through neurological symptoms and signs. The primary mode of diagnosis is testing for NfLs, or neurofilament light chain, which are a protein that indicates the degree of nerve damage.
But what does this have to do with EBV? What even is EBV?
Hold on, Emmy. First, we need to talk about viruses. So, viruses like EBV have DNA that they insert into the cell's nucleus, and the cell can either immediately start translating those DNA genes into RNA that will eventually be made into proteins, or sometimes it lays dormant for a while before it starts making viral proteins in a cycle called the lysogenic cycle.
Guys, I'm still waiting for someone to tell me what EBV is. What's this? I'm the one who has to explain EBV? Okay. The Epstein Barr virus, or EBV, is a disease in the herpes family and is known by scientists as human herpes virus 4. EBV is a double-stranded DNA virus that affects these cells and immortalizes them by preventing apoptosis. I'm practically an expert on this now. Epstein Barr virus transmission occurs through various bodily fluids. Most of us have contracted Epstein Barr virus at some point in our lives.
That's scary.
About 9 out of 10 adults have antibodies that show they have a current or past EBV infection. But what does it have to do with that study that Aaron mentioned earlier?
Well, let's ask her.
Hello, I am scientist Choi, and I'm here today to introduce you to a study that focuses on the relationship between the EBV virus and the MS disease, because there's definitely something fishy going on. The study was conducted over a span of 20 years, and the subjects consisted of 10 million young adults active in the US military. The large number of people helped scientists achieve a more accurate result. In order to measure the onset of MS disease, scientists collected a natural biomarker called SNFLs, or seroneurofilament light chains. They took 3 of these samples, one at the beginning of the experiment, one in the middle, and one at the end. Each MS case was compared to a similar case of MS in order to limit variability and prevent any confounding variables to be at play.
Here are some general results. MS is a rare disease, so only 955 people were diagnosed while serving their time in the military. EBV is common, so how could they be related? The scientists found out that there was a 32-fold increased chance of getting MS after being infected by EBV. On the other hand, the chance of getting MS did not change by infection of other viruses.
One of those viruses is none other than one of EBV's close relatives, the cytomegalovirus, also known as CMV. CMV is another herpes virus that shares many similarities with EBV. CMV contains linear double-stranded DNA and is transmitted via organ transplant, contaminated blood, saliva, and breastfeeding. Like EBV, CMV has severe consequences which can lead to blindness, deafness, and limb disease. However, they do not relate when it comes to MS. Those who are CMV positive had a normal rate of seroconversion of about 57%, or the rate from changing from a negative test to a positive test based on the number of antibodies against the virus found in the body. As the graph shows, EBV increased this rate up to 97%. Despite their similarities, the risk for MS decreased for those who are CMV positive, proving that CMV cannot cause MS. But what about the other herpesviruses?
In 30 MS cases versus 30 people who didn't have MS, researchers looked for an antibody response to test the notion that being infected with MS increases the chance for infection with other viruses. In all the cases, antibody response was similar for every virus except EBV. In EBV, more protein for EBV was detected in those with MS.
Disagreement!
In other words, correlation does not always imply causation. For example, the researchers wondered, could some other variable be causing the increase in risk for both MS and EBV? That was unlikely, because any other factor would have to increase the risk for both MS and EBV by 60 times. The idea of reverse causation was also considered. Could being infected with MS increase the risk of being infected with EBV? That was also unlikely, because antibodies for EBV were present before the rise in SNFL levels. If EBV does in fact cause MS, this means that future treatments for MS can focus on targeting EBV directly.